In high-intensity exercises, there are two different anaerobic metabolisms that help your muscles contract. ATP-PCR and glycolysis are these two systems that are universal for everyone throughout the globe.
PCR is an anaerobic energy, which follows very few steps. Adenosine is connected with 3 phosphates to begin, showing high-energy phosphate bonds. Hydrolysis will then separate the last phosphate from the group, which creates the adenosine triphosphate, ATP phase, which energy is released. Creatine kinase assists with the act of releasing the energy from the PCR. The creatine kinase helps with separating the inorganic phosphate from the creatine. When this happens, the energy that was just released can be used to add the phosphate molecule to an ADP molecule, forming the ATP. This step is called resynthesis.
Some important facts to find from ATP-PCR is that creatine kinase is the rate-limiting enzyme, creating a negative feedback system. This system is a very fast energy production and is stored within the muscles. ATP yield is 1 mole used versus 1 mole created. Because of its fast ATP production, it only lasts from 3 seconds to 15 seconds, with a 3 to 5 minute recovery time. Now that we know how ATP-PCR works, we're now going to discuss glycolysis, which is the second anaerobic metabolism offered in the body. Glycolysis can start at two different forms, as a singular glucose molecule or a chain of glucose molecules called glycogen. For a single glucose, ATP gives off an inorganic phosphate by ATPase to attach and create glucose 6-phosphate. When glycogen breaks down to a single glucose molecule, a phosphate group is already attached, naming it glucose 1-phosphate. Hexokinase rearranges glucose 1-phosphate to also become glucose 6-phosphate. Glucose 6-phosphate is then rearranged by an enzyme called isomerase to leave an open binding site in a 5-carbon ring, which is called fructose 6-phosphate. Another phosphate group is derived from ATP to make fructose 1,6-biphosphate. Fructose 1,6-biphosphate is then lysed by a hydrogenase to complete the halved bonds of fructose. NAD+ floods the fructose and then turns into NADH+, and taking the hydrogen ion to the electron transport chain. ADP removes the phosphate off of each half of the glycerate molecules, creating 2 ATP while also giving off 2 H2O molecules. Another ADP takes the second group of phosphate, which then leaves behind the final product of 2 pyruvate molecules. After the pyruvate has been formed from glycolysis, if there is enough oxygen within the body system, it will go to Krebs cycle to complete oxidative phosphorylation. If there's not enough oxygen within the system, it then turns into a thing we call lactate. Some final thoughts on glycolysis is that PFK is the rate-limiting enzyme, which also has a negative feedback system. Glycolysis is also a fast process that produces 3 ATP per glycogen molecule and 2 ATP per glucose molecule. Glycolysis works from 15 seconds to 2 minutes, with a 2-hour recovery time. Both of these systems are used for high-intensity workouts all across the globe, whether it be from throwing that final punch to sprinting the 100-meter race.